Hello everyone. Today I am going to present on glycolysis, which is the first and most important pathway of carbohydrate metabolism. Glycolysis is the process by which one molecule of glucose is broken down into two molecules of pyruvate, releasing energy in the form of ATP. This pathway occurs in the cytoplasm of all living cells and does not require oxygen directly. It is the common pathway for both aerobic and anaerobic respiration. The term glycolysis is derived from two Greek words, glyco, which means sugar, and lysis, which means breakdown. Therefore, glycolysis means the breakdown of glucose. Before discussing the steps of glycolysis, it is important to understand some basic terms. ATP, adenosine triphosphate. ATP is known as energy currency of the cells. It stores and supplies energy required for cellular activities. When ATP loses one phosphate group, energy is released. Next, ADP, adenosine diphosphate. ADP is a lower energy molecule formed when ATP releases one phosphate group. ADP can again combine with phosphate to form ATP. Next, NAD+, nicotinamide adenine dinucleotide. NAD+ is a coenzyme that acts as an electron and enzyme carrier during metabolic reactions. It accepts hydrogen ions and electrons from substrates. NADH. When NAD plus accepts hydrogen and electrons, it is reduced to NADH. NADH stores high energy electrons and later helps in ATP production through cellular respiration. Phases of glycolysis. Glycolysis consists of 10 enzymatic reactions that occur in two major phases. Phase 1, energy investment phase, also known as preparatory phase. This phase includes steps 1 to 5. During this phase, the cell uses ATP to activate glucose and prepare it for breakdown. In this phase, glucose, which is a 6-carbon molecule, is phosphorylated and modified. Here, two ATP molecules are consumed. One 6-carbon glucose is converted into two 3-carbon molecules. Why it is called the energy investment phase? Just like investing money to start a business, the cell spends energy, which means ATP, initially to obtain more energy later. And here, two ATP molecules are used. Phase 2, energy payoff phase, which is also known as energy generation phase. This phase includes steps from 6 to 10. During this phase, the two 3-carbon molecules are converted into pyruvate. In this phase, 4 ATP and 2 NADH are produced. Two pyruvate molecules are formed. The overall result of glycolysis is the formation of two pyruvate molecules, two NADH molecules, and a net gain of two ATP molecules per glucose molecule. Now, let us discuss the glycolytic pathway step by step. Step 1, phosphorylation of glucose. After understanding the basics of glycolysis, the first reaction in the pathway is the phosphorylation of glucose. In this step, the glucose molecule receives a phosphate group PO4 3- from ATP. The phosphate group is attached to the 6th carbon of glucose, converting it into glucose 6-phosphate, which means G6P. This reaction is catalyzed by the enzyme called hexokinase. Step 2, isomerization of glucose 6-phosphate. The second step of glycolysis involves the conversion of glucose 6-phosphate into fructose 6-phosphate. This reaction is catalyzed by the enzyme phosphoglucose isomerase. It is called an isomerization reaction because the molecular formula remains the same, but the arrangement of atoms within the molecule changes. And in this step, the aldehyde group of glucose 6-phosphate is rearranged to form the ketone group of fructose 6-phosphate. The fructose 6-phosphate is changed from glucose 6-phosphate. The phosphate group remains attached to the sixth carbon and the molecule still contains 6 carbon atoms. No ATP is consumed or produced in this step. Step 3. Phosphorylation of fructose 6-phosphate. The third step of glycolysis is another phosphorylation reaction in which fructose 6-phosphate is converted into fructose 1,6-bisphosphate. The reaction is catalyzed by the enzyme phosphofructokinase 1. In this step, one phosphate group from ATP is added to the first carbon of fructose 6-phosphate. As a result, the molecule now contains two phosphate groups, one at C1 and another at C6, while the carbon skeleton remains unchanged with 6 carbon atoms. This reaction requires one ATP molecule which is converted into ADP. Therefore, it is part of the enzyme investment phase of glycolysis. This is an important, irreversible and rate-limiting step of glycolysis. Phosphofructokinase 1 regulates the flow of glucose through glycolysis and the formation of fructose 1,6-bisphosphate commits the molecule to continue through the glycolytic pathway. Step 4. Cleavage of fructose 1,6-bisphosphate. The fourth step of glycolysis is a cleavage reaction where the 6-carbon molecule fructose 1,6-bisphosphate is split into two 3-carbon molecules. The enzyme aldolase catalyzes this reaction. The molecule is cleaved between the C3 and C4 carbon atoms, producing glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This step is important because it converts the single 6-carbon molecule into two smaller 3-carbon molecules that can undergo further reactions in glycolysis. G3P can directly proceed to the next step, whereas DHAP is converted into G3P in step 5. Therefore, from step 6 onwards, the reactions of glycolysis occur twice for every glucose molecule. And the step 5 is isomerization of DHAP. The fifth step of glycolysis is an isomerase reaction in which dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. This reaction is catalyzed by the enzyme triose phosphate isomerase. In step 4, two 3-carbon molecules were formed, G3P and DHAP. And step 5, as only G3P can directly enter the next stage of glycolysis. DHAP is rearranged into G3P so that both 3-carbon molecules can continue through the remaining steps. Step 6. Oxidation and phosphorylation of glyceraldehyde 3-phosphate. In the sixth step of glycolysis, glyceraldehyde 3-phosphate undergoes oxidation and phosphorylation to form 1,3-bisphosphoglycerate. The reaction is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. During this reaction, G3P loses hydrogen and high-energy electrons which are accepted by NAD+, resulting in the formation of NADH. NAD+ accepts electron and hydrogen during oxidation and gets reduced to NADH. An additional hydrogen remains in the reaction medium. At the same time, an inorganic phosphate is added to G3P to form 1,3-BP3G. Unlike the phosphorylation reactions in step 1 and 3, ATP is not used here. The energy released during oxidation provides the energy required for the addition of the phosphate group. Since one glucose molecule produces two G3P molecules, this reaction occurs twice and produces two NADH molecules per glucose molecule. Step 7. Formation of ATP by substrate level phosphorylation. In the seventh step of glycolysis, 1,3-bisphosphoglycerate is converted into 3-phosphoglycerate. During this reaction, the high-energy phosphate group present in 1,3-BPG is transferred directly to ADP, resulting in the formation of ATP. The reaction is catalyzed by the enzyme phosphoglycerate kinase. This is the first ATP producing step of glycolysis and is called substrate level phosphorylation because ATP is formed by the direct transfer of phosphate group from a high energy substrate to ADP. Since one glucose molecule produces two molecules of 1,3-BPG, this reaction occurs twice and produces two ATP molecules per glucose molecule. Step 8. Rearrangement of 3-phosphoglycerate. The eighth step of glycolysis is an isomerization or rearrangement reaction. In this step, 3-phosphoglycerate is converted into 2-phosphoglycerate by the enzyme phosphoglycerate mutase. The main change in this reaction is the movement of the phosphate group from the third carbon to the second carbon. The carbon skeleton remains unchanged and no ATP is consumed or produced here. This rearrangement is important because it prepares the molecule for the next step of glycolysis. The position of the phosphate group allows 2-phosphoglycerate to undergo dehydration in step 9, resulting in the formation of phosphoenol pyruvate, a high energy compound that will be used to generate ATP in the final step. Step 9. Dehydration of 2-phosphoglycerate. In the ninth step of glycolysis, 2-phosphoglycerate is converted into phosphoenol pyruvate by the dehydration reaction. The enzyme involved is enolase. In this reaction, a water molecule is removed from 2-phosphoglycerate. The removal of water causes a rearrangement of the molecule and results in the formation of PEP, which is a high energy phosphate compound. Step 10. Formation of pyruvate and ATP. The 10th and final step of glycolysis is the conversion of phosphoenol pyruvate into pyruvate. This reaction is catalyzed by the enzyme pyruvate kinase. PEP is a high-energy 3-carbon compound formed in step 9. In this step, PEP transfers its high-energy phosphate group directly to ADP, resulting in the formation of ATP. The remaining 3-carbon molecule is converted into pyruvate. Since ATP is produced by the direct transfer of a phosphate group from the substrate PEP to ADP. This process is called substrate level phosphorylation. As one glucose molecule produces two PEP molecules, this reaction occurs twice for each glucose molecule. Therefore, two ATP and two pyruvate molecules are produced in step 10. This step is irreversible under cellular conditions and marks the completion of glycolysis. The pyruvate formed can subsequently enter different metabolic pathways depending on the availability of oxygen. In this slide, we can see the complete glycolytic pathway consisting of 10 enzyme catalyst reactions. Glycolysis occurs in the cytoplasm and converts one molecule of glucose into two molecules of pyruvate with the production of ATP and NADH. The pathway begins with glucose, a 6-carbon molecule, in which the first step is glucose is phosphorylated by the enzyme called hexokinase using one ATP molecule to form glucose 6-phosphate. This phosphorylation traps glucose inside the cell. In step 2, glucose 6-phosphate is rearranged into fructose 6-phosphate by phosphoglucose isomerase. In step 3, another ATP molecule is utilized by phosphofructokinase 1 to convert fructose 6-phosphate into fructose 1,6-bisphosphate. This is the rate limiting and committed step of glycolysis. In step 4, the 6-carbon fructose 1,6-bisphosphate is split by aldolase into two 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. In step 5, DHAP is converted into another molecule of glyceraldehyde 3-phosphate. At this point, two molecules of G3P are available. The first 5 steps constitute the energy investment phase, during which two ATP molecules are consumed. The next 5 steps form the energy payoff phase. In step 6, glyceraldehyde 3-phosphate is oxidized and NAD plus is reduced to NADH, producing 1,3-bisphosphoglycerate. Since there are two G3P molecules, two NADH molecules are formed. In step 7, the high energy phosphate of 1,3-bisphosphate glycerate is transferred to ADP to form ATP. This is the first ATP generating steps of glycolysis. In step 8, 3-phosphoglycerate is rearranged into 2-phosphoglycerate. In step 9, a water molecule is removed by enolase to form phosphoenol pyruvate or PEP, which is a high energy compound. Finally, in step 10, PEP transfers its phosphate group to ADP through the action of pyruvate kinase, producing ATP and forming pyruvate. Since two PEP molecules are present, two ATP molecules and two pyruvate molecules are produced. Overall, one molecule of glucose enters the glycolytic pathway and passes through these 10 enzyme catalyst reactions. In the first 5 steps, the cell inverts energy by utilizing two molecules to activate and prepare glucose for breakdown. The 6-carbon glucose molecule is then converted into two 3-carbon G3P molecules. In the second half of glycolysis, these two G3P molecules undergo oxidation and several rearrangement reactions, resulting in the formation of ATP, NADH, and finally pyruvate. At the end of glycolysis, one glucose molecule produces two molecules of pyruvate, four ATP molecules are generated in total. But since two ATP molecules were used during the energy investment phase, the net gain is two ATP molecules. In addition, two NADH molecules are produced which carry high energy electrons for further energy production. Therefore, the outcome of glycolysis is one glucose molecule gives two pyruvate, two NADH, and a net gain of two ATP molecules. The pyruvate formed at the end of glycolysis does not mean that energy metabolism ends here. Depending on the availability of oxygen and the cellular conditions, pyruvate can either enter further aerobic metabolism through conversion to acetyl-CoA TCA cycle or undergo fermentation under anaerobic conditions. Thus, glycolysis acts as an important link between glucose breakdown and the subsequent pathways of cellular energy metabolism. So, in a simple way, we can understand glycolysis as the breakdown of one 6-carbon glucose molecule into two 3-carbon pyruvate molecules through 10 enzymes catalyst reactions. During the first 5 steps, the cell invests to ATP to activate the glucose molecule. In the next 5 steps, energy is recovered in the form of ATP and NADH. Finally, glycolysis gives 2 pyruvate, 2 NADH, and a net gain of 2 ATP. The pyruvate can enter further metabolic pathways depending on the availability of oxygen. Thus, glycolysis is an important pathway that provides energy to the cell and serves as the starting point for further glucose metabolism. So I hope you now have a clear understanding of the glycolysis pathway and how glucose is utilized by our cells to generate energy. We have seen how the glucose pathway progresses through different enzymatic reactions and how each step has its own importance in the overall process of carbohydrate metabolism. I hope this presentation has made the concept of glycolysis easier to understand and remember, especially the role of ATP, ADP, NAD+, and NADH and the different enzymes involved in the pathway. Glycolysis may look like a complex pathway with 10 different steps, but once we understand what happens to the carbon skeleton and how energy is invested and produced, the entire pathway becomes much easier to follow. I hope this presentation has given you a better understanding of the topic and I will help you connect glycolysis with the other pathways of cellular respiration. With this, I would like to conclude my presentation on glycolysis. Thank you everyone for your patient listening and valuable time.